My name is Dr. Len Cinto. I'm a professor in the Department of Earth and Environment and a research scientist with the Institute of Environment at Florida International University. We're currently in the freshwater biogeochemistry lab. We work in a number of aquatic environments, including freshwater systems and how freshwater terrestrial systems impact our coastal marine environments. We're here to talk about a project called the Identification of Lake Worth Lagoon Pollutant Load Source Areas and the Efficacy of Surface Water Management Upgrades Designed to Improve Water Quality. Basically, it's a long name for what we call the Lake Worth Lagoon Project. The principal investigator is my colleague, Dr. Randall Parkinson. I'm a co-PI, Len Cinto, along with other co-PIs, Dr. Piero Gardinali of the Department of Chemistry and Biochemistry here at FIU, and Dr. Melania Secco-Pieri, who is currently a colleague in Brazil. I'm joined here today with two of my colleagues on the project, Dr. Lesmes Jerez and the PhD candidate student, Mr. Oluwatosin Orimolade. Olu. This project is funded by the U.S. Environmental Protection Agency, Region 4, the South Florida program. This project that we call the Lake Worth Lagoon Project is important for a number of reasons. One, we have a number of impacted water bodies in South Florida. Due to agricultural and urban development, we've loaded our canal systems with a number of contaminants, including excess nitrogen and phosphorus, which ultimately make their way from the terrestrial environment into our coastal marine environments. And therefore, the impact kind of moves downstream to affect the downstream environment. Lake Worth Lagoon has been a highly impacted water body. But the problem doesn't start in the lagoon. It starts in the urban environment, the urban and agricultural environment. So what we're looking at is areas and land uses in this watershed that are the sources of these contaminants. And looking for sources and sinks. Where in this environment can some of this material be sequestered, buried up as sediments, and therefore at least temporarily removed from the short-term cycling of nutrients and impacts in downstream water quality. How do we manage this water so that we could maintain our flood control and water movement without exacerbating the downstream export of contaminants, right? So we want to kind of keep the contaminants in the terrestrial environment, but allow the water to pass through. So how do people do that? Well, they put in a bunch of best management practices, operational procedures like flow rates, structure openings, things like that, fertilizer limits in certain areas. All these BMPs are best management practices. But should these things be applied generally, or should they specifically be targeted at those areas that are creating the greatest sources? And that's what we're trying to do here. We're looking to see where in the system are these contaminants sourced? How do they transfer through the canals? And what is their ultimate sink in Lake Worth Lagoon and their ultimate effects?
What I do basically on this project is that I look at how flow imparts nutrient transport. What we do is go to the field, collect water samples, sediment samples, and in our water sample, we try to analyze for total suspended solids. And we also measure the flow in the water system. We want to see how this flow imparts the transport of nutrients downstream. When we come back to field, we try to tie the result of our data sample to our flow to see the relationship. It is important to know the flow data in canals because the canals are not natural way through which water is transported. So it is being managed. And we want to look at how this management help in the release of water and also the release of nutrients that is in this water. We collect water samples and sediments across flow pattern from Lake Okeechobee. I will bring those samples to the lab. We analyze them for a range of nutrients like phosphorus and nitrogen and trace metals. For the flow data, we use the M9ADCP device to collect the discharge or the velocity of the water at that time when we also collect our water samples and sediment. So when the results is back, we tie the results of the water to the flow to be able to understand how flow imparts the transport of nutrients and the mass of nutrient being exported.
It's really important to know the physical-chemical characteristics in water, solids, and sediments from the upstream to the downstream of this system, because we really need to know exactly how much pollutants are coming from the Lake Okeechobee and how much pollutants are actually being delivered at the downstream environment, in this case at the Lake Worth Lagoon. But it's not only about what is entering and what is going out, but also what is happening in between. What is the gradient of change of those pollutants? What is the fate and transport of those contaminants through these canal systems? This is really, really important, because that it is going to help us to understand how those water systems are working. And this is also very important because we are not only advancing the science, but also improving the management by the people and for the people. For example, it's very important to understand how we can operate the flow, and how controlling the flow is not only about inflowing, but also the exporting of contaminants from the upstream through the downstream environment. We are collecting all these samples by doing extensive field campaigns that involves to collect water, solids, and sediment samples using multiple equipments and devices from boats and from established bridges in order to collect those samples. That requires a lot of effort, but it's actually fun, and we have a great time doing this process.